adult life is important to know. And also pharyngeal arches, what are the arch derivatives and what are the associated anomalies. These are some very, very important topic and must-do topic not to miss before your exams. Going on with some tips and tricks about this module, you have to know the timing of the developmental changes. For example, when is the bilaminar disc formed? When is the trilaminar disc formed? These are some important developmental changes, timings that you have to know. Always try to have a pictographic memory for this module because you can expect image-based questions. Commonly asked are the images of a Graafian follicle, the images of a sperm fusing with an ovum, etc. And use mnemonics for easy memory, especially when you have topics like the pharyngeal arch derivatives. It's always a good idea to have some mnemonics so that you can remember them easily. Clinical application. We know embryology is quite a dry topic for most of us because we're not following that in our clinical day-to-day practice. But however, try to have a clinical implications behind each and everything that you're going to study. For example, the urogenital abnormalities, think about the congenital malformation of the uterus and try to associate with them. Then the topics get really interesting to study. And also you understand why the RCOG is focusing on these topics. The library that we provide you is always a treasure. Whatever you ask is there for you. And have a look at the summaries and the important videos that we have presented there in the library for you guys. So let's start with the discussion of the development of the urogenital system. So to start with, we need to understand there are three different structures that are formed initially. They are the pronephros, the mesonephros, and the metanephros. So each of them have these ducts and tubules, and each of them has a particular relevance. So these are the terminologies to start off knowing about it. So in a nutshell, I have tried to summarize the development of kidney for easy understanding and memorizing. So to start off, we need to understand that the urogenital system develops from the intermediate mesoderm. So the first structure to form here is from the cervical intermediate mesoderm, and this is called as the pronephric duct and tubules. They appear and disappear by about 4th week. So essentially, they are non-functional, they just degenerate and they provide a roadmap for the future development of the kidney. Coming next in development is the mesonephric duct and tubules, which originate from the thoracic and the lumbar intermediate mesoderm. They start to appear by the 5th week and they function as the primary excretory organ till the 9th week of gestation. The complete degeneration is seen by around 12 weeks. So knowing the importance of this mesonephric duct and tubules, the tubules are the one which act as the temporary kidneys till the permanent ones are formed. The mesonephric duct degenerates in the females, whereas in males, the function shifts from the urinary system to the genital system. It is known as the Wolffian duct and it forms the epididymis, the vas deferens and the ejaculatory duct. Coming to the ureteric bud, which is another important terminology that we have to remember here. So we need to talk about the metanephros first. Why did I mention about ureteric bud? Because the ureteric bud is actually a derivative of the mesonephric duct. So from here, it's going into the metanephros, and the metanephros is the one that is going to form the kidney per se. So what is the role of ureteric bud? This ureteric bud, as I mentioned, it is a derivative of the mesonephric duct. This goes on to form the collecting tubules, the calyces, and the ureter. And when this ureteric bud encroaches into the metanephros, it is going to bring in differentiation there. So coming to metanephros, this arises from the lumbar and the sacral intermediate mesoderm. They appear by around 5 weeks of gestation and they become functional by about 10 weeks of gestation. So it is from this metanephric blastema, we have the metanephrogenic cap that is formed, and this is going to form the nephron per se, which is constituting of the Bowman's capsule, the proximal convoluted tubule, the loop of Henle, and the distal convoluted tubule. At the end of all this, we also need to understand the cloaca and the differentiation of the cloaca, because that is going to help us understand the development of the urinary bladder, which is another major component of the urogenital system. So what is cloaca? Cloaca is a structure that is present in the caudalmost end of the embryo. And here we have the endoderm and the ectoderm fused together with no mesoderm in between. And when this is a membranous structure, and when this cloaca gets divided, we have the formation of the rectum and the primitive urogenital sinus. It is from this primitive urogenital sinus we have development of the urinary bladder, the primitive urethra also. So what happens in this primitive urogenital sinus? This is divided into three different parts. The three different parts constitute the cranial-most one, which is called as the vesical part, where most of the bladder is developing from here. The second part is the middle part or the pelvic part, which is going to form the main part of the male urethra, and the entire part of the female urethra forms from here. The last part of the urogenital sinus is called as the phallic part or the caudal part, and this is going to grow towards the genital tubercle. So this is the importance of knowing about the urogenital sinus. So as we understand the development of the bladder, the main area is from the urogenital sinus. So what are the important key points that you need to brush up before your exam? We have the intermediate mesoderm, which is a potential SBA. And from the intermediate mesoderm, we have the pronephros, the mesonephros, and the metanephros. And from the cloaca, we know the urogenital sinus is going to come. So the permanent urinary system, the kidneys are formed from the metanephros. The upper urinary tract, that is the ureter, the calyces, and the collecting tubules are formed from the mesonephros, and the lower urinary tract, that which constitutes of the bladder and the urethra, are developing from the urogenital sinus. So these are some important key points not to miss in the development of the urinary system. Coming to the genital system development, first we need to understand that initially these gonads are indifferent. That means there's no differentiation into the male and the female gonads. So the development of either the testis or the ovary has these two different stages, the indifferent stage and the different stage. In the indifferent stage, which is seen before 7 weeks, and then the differentiation starts after 7 weeks, where this gonad gets converted into either a testis or an ovary. So each of these gonads develop from essentially three different structures. What are they? First one is the genital ridge. What is the genital ridge? It is the medial part of the mesonephros, and this is going to give formation to the capsule, the stroma, and the connective tissue of the gonad. Second in number is the coelomic epithelium. The coelomic epithelium is going to cover the genital ridge, and it is from this where the Sertoli cells of the testis or the follicular cells of the ovary are going to form. Last but not the least, the most important one is the primordial germ cells. These primordial germ cells originate from the wall of the yolk sac, and they migrate all the way to the posterior part of the embryo. And it is these cells which are going to give rise to either the spermatogonia or the oogonia respectively. So this is what happens initially. We have the undifferentiated gonad. And what brings about this undifferentiation into the differentiation? Well, it is the presence of the SRY gene. The SRY gene is the male determining factor. And where is this present? This is present in the sex determining region of the Y chromosome. This is important. So in the short arm of the Y chromosome, we have the SRY gene. And this particular gene is going to help in the development of the male gonadal system. And absence of this leads to the development of the female gonadal system. The other really important points to remember in the gonadal system, well, we do know there are many things, but the key important ones that you have to remember for exam are the homologues. What are the different male and female homologues? It corresponds to both the internal structures as well as the external genitalia. Now coming to the male, the main duct which is going to form the entire male system is called as the Wolffian duct. The female duct is called as the Mullerian duct or the paramesonephric duct. The Wolffian duct is going to form the ductus epididymis, the vas deferens, the seminal vesicle, the ureter, the renal pelvis, calyces, and the collecting ducts. The other name of the Wolffian duct is the mesonephric duct. In the female, we understand that it has a urinary system role. That means this is going to form the ureter, the renal pelvis, calyces, as well as the collecting ducts. However, coming to the genital system, it is considered vestigial. So it is only going to form the Gardner's duct, which is also called as appendix vesiculosa. The duct that is of importance for the females is the Mullerian duct or the paramesonephric duct, which is going to form the major part of the female genital system. That is, it is going to form the uterus, the fallopian tubes, and also part of vagina. Whereas in the males, this is going to form the appendix testis and the utriculus prostaticus. These two are considered as vestigial organs in the males. So the questions in exam can be targeted as asking for the homologues or asking for the development of these structures, which can either come as a direct SBA or you can have image-based questions on these topics. Coming to the urogenital sinus, we know it is important because it's going to form the bladder and urethra. Apart from that, the prostate develops in the males, and it is also mentioned in few literature that the caudal part of vagina can develop from the urogenital system. However, this is an area of debate. Coming to the tubules of the mesonephros, this is vestigial in females, forming the epiphuron and the paraphuron, whereas in males, it's going to form the ductili efferens and the paradydymus. So this is all about the homologues of the internal structures in the male and the female. It's important to understand what are the homologues and also where is the embryological origin of these structures. Coming to another area of importance is understanding the development of the external genitalia. Again, I've made it as a summary for easy and better understanding for the exam point of view. The external genitalia, also as the internal genitalia, stands undifferentiated, and then later on it gets differentiated into the male and female structures. So the embryological origin of the genital tubercle, which is going to get influenced under the hormones, and in a male it is going to form the glans penis. The structure that is homologues to this is the glans clitoris in the females. It also forms the corpus cavernosum and the spongiosum in the males, whereas in females, the homologue is the vestibular bulbs. Coming to the urogenital sinus, it is going to form the bulbourethral gland or the Cowper's gland in males. The homologue is the Bartholin gland or the greater vestibular glands in the females. The other structure, it forms the prostate gland in the males, and the homologue is the Skene's gland or the urethral and the paraurethral glands in females. So this question can either be targeted at what is the embryological point of origin or the homologues between the male and the female. Both can be expected in exam. Coming to the urogenital folds, the urogenital fold is going to form the labia minora in females. The homologue to this is going to be the ventral shaft of penis or the penile urethra in males. Coming to the labioscrotal swelling, this is going to form the labia majora in the females, and the homologous structure is the scrotum in the males. So understanding this external genitalia is always considered important because many times we have seen repeated questions being targeted in and around this area. So let us try to understand some important questions, and also let us see how we are able to answer them with all the information that was provided right now. So question number 1, when does the metanephros become functional? So this is where I mentioned that you have to know some kind of a timeline development of the embryo too. So the options provided here are 3 weeks of development, 4 weeks of development, week 10, just before birth or just after birth. We know that metanephros are the ones which are going to form the kidney per se. So kidneys do become functional when the fetus is already inside, and it is by about 10 weeks of development. So the beginning starts by around week 5, and by 10 weeks, it becomes completely functional. Pronephros is completely non-functional in humans. How is the trigone of the posterior wall of the urinary bladder formed? We always associate the formation of urinary bladder with the urogenital sinus, but however, we need to understand that there's a difference in the development of trigone, which is present in the posterior wall of the urinary bladder. So what are the options we have for this? One is the incorporation of the lower ends of the mesonephric duct or is it incorporation of the lower ends of the pronephric duct, or is it from the metanephric blastema, or the mesonephric tubules, or the pronephric tubules? So we know the pronephros is completely non-functional, so the probability of the pronephros here being very less. So we are left off with options of mesonephros and metanephros. We need to understand that metanephros is always associated with the kidney per se, that is the development of the nephron. So we have two more options left behind, which is the mesonephric duct or mesonephric tubule. So the answer for this is the mesonephric duct. Mesonephric duct is going to contribute to the connective tissue component of the posterior wall of the trigone. The transitional epithelium, which is going to line the entire bladder, including the trigone, is of endodermal in origin. Whereas we saw the major urogenital system is originating from the intermediate mesoderm. So again, there's a difference that is seen in the trigone. When does the indifferent embryo complete the phenotypic sexual differentiation? So I had purposefully put this question here because we need to understand there's a difference between the phenotypic sexual differentiation and the genotypic sexual differentiation. So phenotypic, let's look at the options. Week 3, week 5, week 7, week 12, or week 20. The answer is week 20 of development. We understand that before 7 weeks, it's completely an indifferent gonad, and from 7 weeks, the differentiation starts. However, by week 12, the male and the female characteristics can be recognized, but only by week 20, the phenotypic sexual differentiation is complete. So to make it out in the scan, it takes 20 weeks of development. Labia majora in the female is homologous to which structure in the male? It's an easy one where you need to understand the homologues between the male and the female and also the embryological origin. So have the tabular column in your mind. So the labia majora is homologous to, is it with the penis or the gubernaculum testis, scrotum, the tunica or the glans penis? The answer for labia majora is the scrotum. And this is a clinically applicable question. We have a particular picture that's been presented to us, and we have been asked to identify what is the congenital malformation. The options that are presented here are, is it a biconvate uterus? Is it a uniconvate uterus? Is it a uterine didelphys? Is it a septate uterus? Or is it an arcuate uterus? These kind of questions can be a little tricky if you do not know how these images actually look like. So the ideal thing for us to remember before the exam is to just have a quick look at how the congenital malformation of the uterus may appear. And also remember, you can expect an image-based question out of these topics. So any topic in embryology which has a clinical relevance, please do have a look at the images before the exam. That might really help you for the last minute preparation and revision too. So let's look into the image that's been presented here. So from what we understand, we see that there are 2 ovaries, 2 fallopian tubes, and there are 2 uterus. Coming down to the cervix, we are able to identify there are 2 different cervical openings. There are 2 different internal and external os, which is finally we have 1 vagina where the cervical os actually opens into. So this is a complete duplication of the uterus, including the cervix. So this is called as a didelphys uterus. So the most common thing that we confuse with the didelphys uterus is a biconvate uterus. So the basic difference between a biconvate uterus and a didelphys uterus is that in didelphys we also have duplication of the cervix, whereas in biconvate uterus we only have a single cervix that is there. In uniconvate uterus, there is only 1 horn of the uterus that's developed. So let me just draw a small picture here. So in uniconvate uterus, we only have 1 horn of the uterus that is developed and the other horn is completely missing. Okay. Sometimes what happens, there can be a very small rudimentary horn and this can be separate or this can be communicating with this horn. So based on this, we have different types of uniconvate uterus. We have a rudimentary horn, communicating and non-communicating. In biconvate uterus, it is usually 2 different uterus like this. but the cervix is usually 1. Now again, coming to the difference between a septate uterus and a biconvate uterus. So the difference is, in the biconvate uterus, if we look outside, there is a definite dimpling which is seen in the fundus. Whereas in the septate uterus, the fundal dimpling is usually not seen. However, in the center, we find that there is a septum that is extending from the fundus down below, and this septum can either be a partially dividing septum or a completely dividing septum. So these are all the different types of images that you can expect in exam. So that's all about this, because the congenital malformation of the uterus is actually considered as a very, very important topic for exam. So these are some really, really important topics that you should not miss in embryology, especially in the urogenital system. So for those of you who are doing your last minute revisions, this video is definitely going to be helpful, because we have covered many, many important topics and important points too, which can come as potential SBAs for your exams. So wishing all of you all the very best for your exams, and keep watching us and subscribe us on YouTube. Thank you.
Recording stopped.